Hello my dear students, I am Roshni from Learner Hub, the free learning platform, and today I am here with the third video of the video series on Heredity and Evolution class 10th Science, and in this video we are going to learn about homologous and heterologous chromosomes, and we are also going to understand Mendel's experiments results in more detail. So this is going to be a very important video, so please watch it till the end. Let's get started for now. Let us now see what are homologous and heterologous chromosomes. So this is again something new. When we are talking about chromosomes, it is important to know about homologous and heterologous chromosomes. Now, homologous chromosomes, let us discuss first. A set of one maternal chromosome and one paternal chromosome that pair up with each other inside a cell. So what are these homologous chromosomes? Now the word homo itself means something which is similar. So a set of one maternal chromosome, that means the chromosome from mother, and one paternal chromosome, that is the chromosome from father, that pair up each other inside a cell. So as I said, inside a cell, the chromosomes, right now I am talking of diploid cells, because only the diploid cells will have two sets of chromosomes, right? So inside the diploid cell, chromosomes will always exist in pairs. So the pair of chromosomes where one set is from the father, the other set is from the mother, and they are similar to each other. They are known as homologous chromosomes. So they are of same size and shape. Their form will remain the same. So such chromosomes are called homologous chromosomes. They bear corresponding genes governing the same traits. Now we know that when you look at the structure of a chromosome, so on the chromosomes you have genes, right? And these genes actually control the different characters in our body. So when I talk about homologous chromosomes, they are somewhat like this. Let us suppose this is one chromosome, this is another chromosome, right? And here you have the genes located. So such regions, so these region of the two chromosomes are known as homologous regions, which code for the same gene. That means they both code for the same gene. Now you might ask that the same gene is present here also, but what are these two? These are the sister chromatids. I have already explained you, right? These are known as chromatids. This center part which joins the two chromatids is known as centromere. So the homologous chromosomes are two different chromosomes where same genes are present in the homologous region, right? So they have these homologous region basically consist of different version of the same gene, right? So let me take an example. Let us suppose if I talk about the eye color, right? So this is the paternal chromosome. Let us suppose this is the paternal chromosome and this is the maternal chromosome. So if I am talking about eye color, let us suppose that the father's eye color is blue and the mother's eye color is black. So in this case, if this gene corresponds to the trait of eye color, right? So for eye color, both of them will represent brown, both of these. So that means the sister chromatids are exact replica of each other. But these maternal chromosomes, they will represent blue. So these two will again be representing blue. But if you compare this and this is also representing eye color. This is also representing eye color. That means both are representing same gene but different versions. This is representing black color, this is representing blue color. So they are representing different versions of the same gene. So now you understand what are homologous chromosomes. They are two sets of chromosomes which exist in pair, one contributed by father, one contributed by mother. And they both are of same shape and size, and they bear genes governing the same traits in the corresponding region. That means the gene governing eye color are present in the same region. Similarly, let us suppose the gene governing the skin color is present in the same region. Similarly, the gene governing the earlobe shape, that is also present along the same region. So these are known as homologous chromosomes. So now here in this picture also you can see the homologous chromosomes. So here these blue color, here you can see this represents the eye color, this blue color represents the father's eye color, this black color represents the mother eye color. So they are known as homologous chromosomes. Similarly, if I talk about heterologous chromosomes, that means different. So they differ in shape, size and function. Like in case of homologous chromosome, they were of same shape and size. But here they have different shapes, different sizes and different functions as well. Because here the function was also similar. Everything was corresponding to eye color. But here that is not the case. So they do not belong to the same pair. So that means if we take one pair of this chromosome and another pair of some other chromosome, maybe one part is corresponding to eye color and the other part is corresponding to skin color. So those kind of chromosomes are known as heterologous chromosomes, right? So why I am explaining this is because these terms homologous and heterologous will come as we go ahead and discuss about Mendel's experiments. So now we will know about a new concept called allele. So what are allele? So now I already told you the experiment which Mendel had performed. Now how do we denote the traits of different organisms? Like as I said, there is one tall plant, there is one short plant. So how do we denote the characteristics? Now the traits are basically denoted with the help of letters. For example, in order to describe a tall plant, maybe I can denote it by a capital T. In order to denote a trait, that is a dwarf plant, so maybe I can denote it by a small t. So that is how we denote things, right? So now, allele is one member of a pair of genes that occupy a given position on a homologous chromosome, because we saw that when you take the example of the homologous chromosome, so what do we see here? So in this homologous chromosome, this blue color and this dark blue color, they all represent the same gene, right? So but they are present in pairs. This is one pair, this is one member of the pair, this is another member of the pair. So allele is one member of a pair of genes that occupy a given position on a homologous chromosome. So let us suppose if I denote, let me take an example this way. Let us suppose if this denotes the blue eye color, which is denoted by a small b. Let us suppose if this denotes the black eye color, which is denoted by a capital B. So when you combine together these two chromosomes, because they are existing in pairs, so what do you get? Small b, capital B. So what would be the eye color when taken together? It will be black, because black is the dominant trait, right? Now each of these pair, that means this small b and this capital B, they are separately known as an allele. So allele is one member of a pair of genes that occupy a given position on a homologous chromosome. Why they occupy a given position? Because they are going to correspond to the same trait, right? Because in homologous chromosome, if they occupy the corresponding positions, that itself means that they are going to correspond to the same trait. Now let us look at some examples. Let us suppose if we take this example of the dwarf plant. So how do we denote its allele? Now this dwarf plant, the plant is in Mendel's experiment, we saw that dwarf was the recessive trait, right? It was not expressed in F1 generation. Now we have also studied that when a recessive trait gets expressed, when it inherits the trait from both of its parents. So that means this plant is dwarf because it has got a dwarf trait from its both the parents, right? So how do we denote this? We denote it as small t, small t. That means we are talking about a perfectly dwarf plant. It has received a small t from one parent and a small t from the other parent as well. So each t is known as an allele. Similarly, if we are talking about a tall plant, so this tall plant, for a plant to be tall, there are many possibilities. Maybe it has got the tall trait from both the parents, maybe it has got tall trait from one of the parents. So we can represent it as capital T, which shows that it has received a tall trait from the father as well as a tall trait from the mother. Similarly, we can also represent it as capital T small t, that means it has received a tall trait from one parent whereas a dwarf trait from another parent. But since tall trait was dominant, so the plant is tall. Similarly, we can also represent it as small t, capital T, that is it has received a dwarf trait from the first parent and a tall trait from the second parent, right? And each of these members, so we express the characteristic of any organism in terms of, with the help of letters, right? Now each member of this pair is known as an allele. Right, so allele is a term which is new to you, and this is the significance of allele. So let us look at these examples. So these are the examples of alleles. Let us now talk about homozygous and heterozygous organisms. Now that we have spoken about alleles, we also saw that there are many ways by which we can express the alleles or the traits of organisms. So based on what type of alleles are present in an organism, they can be classified into two types, homozygous and heterozygous. Homozygous organisms, the word homo always means something related to similar. So these organisms will have identical alleles, that is similar alleles on homologous chromosomes. For example, capital T, small t. So that means if we talk about a dwarf plant, as I mentioned in the previous slide also, we write it as TT. Since it has received the same traits from both the parents, that is why it is known as a homozygous dwarf plant. Similarly, if we talk about a tall plant which has received tall trait from both the parents, we write it as capital T. So both the alleles are identical. So it is known as a homozygous tall plant. Now, if self-bred gives rise to offsprings with same traits, that means we all know what is self-breeding. Self-breeding is nothing but self-pollination. So, self-pollination means if the pollination happens in the flower of the same plant. So, let us see exactly what happens on self-breeding. Now, let us suppose that if we self-pollinate the homozygous tall plant, that means tall plant with another tall plant. So now this tall plant has two alleles, T and T. Similarly, this tall plant also has two alleles. Now what are the possibilities which are possible? This T can combine with this can also combine with this can combine with this, or this can combine with this. So what do we see? In all the cases, we get homozygous tall plants, right? So all tall homozygous. So if it is self-bred, it gives rise to offsprings with the same traits. That means this is also homozygous tall, so they are also homozygous tall. Similarly, if we self-bred the dwarf plants, in that case also what are the possibilities? This will give out its alleles, and these alleles can combine in any of these fashion. So in all the cases we get homozygous dwarf plants, right? So this is all about an homozygous organism. Let us now look at heterozygous organism. Now I am sure you know by now what is heterozygous. It is organism with different alleles for a character on homologous chromosomes, because both are true for homologous chromosomes, because only for homologous chromosomes both the chromosomes will correspond to the same character, right? So here what is that character which we are talking about? In case of these plants, we are talking about the height. So whether it is tall or dwarf, it is talking about the height of the plant. So examples of heterozygous organisms would be small t capital T or capital T small t. So here we have two different alleles, but since the tall is a dominant trait, so the plant is tall. So if one allele is dominant, the trait will also be dominant. So in case In this case, we can say that these are also known as hybrid. Heterozygous is also called hybrid. So we can call these as hybrid tall plants or heterozygous tall plants. Right now, aren't you curious to know that what would happen if we self-bred these heterozygous tall plants? So let us try to see what will happen if we try to self-bred them. Let us suppose if capital T small t is self-pollinated with capital T small t, what will happen? It has these two alleles, this has these two alleles. So these are the possibilities that can occur, right? So what do we get? In the first option, we'll get capital T, then we'll get capital T, small t, then we'll get small t, capital T, then we'll get small t, small t. So in this case, if you see, the products are not all identical to the parent organism, right? You see that only 2 of them are heterozygous. So 50% are heterozygous. The remaining 2 are homozygous. So we can say that if we self-bred heterozygous organisms, only 50% of them are heterozygous, right? Also, we see that how many of them are tall and how many of them are short. We can say that this is tall, this is also tall. So this is tall homozygous, this is tall hybrid, this is also tall hybrid, and this one is dwarf. So what do we see? The result is 3 out of 4 are tall. That means 75% of them are tall. 1 out of 4 is dwarf. That is 25% of the results are dwarf, right? Okay, so this was all about homozygous and heterozygous organisms. So with this, I think I have covered all the important basics which was needed to understand the Mendel's experiments in detail. So let us have a quick review. What are the things that we studied? We talked about what happens during sexual reproduction. How does the chromosome numbers change? We talked about the diploid and haploid cells. We also talked about homologous, heterologous chromosomes, homozygous and heterozygous organisms, and alleles, right? So with this basic knowledge, I think we are good to go with understanding Mendel's experiment. Now let us look at Mendel's experiment in little more detail in terms of the alleles, in terms of homozygous and heterozygous. So now we can use those terms and try to understand Mendel's experiment. So what was Mendel's first? Mendel actually performed 2 sets of experiment. So the experiment which I had mentioned earlier, that was his first experiment. So we will get back to his first experiment once again. What he did, he took homozygous tall plants. So now you know what are homozygous tall plants. If they are homozygous, that means both the alleles will be same. If they are tall, so it will be capital T. So capital T, capital T. And he also took homozygous dwarf plants. That is small t, small t. So this is what he took. And then he cross-pollinated them. Right? So now on cross-pollination, what was the result? So on cross-pollination, it was seen that all the plants which were obtained were all tall. So now I think we are capable of understanding how all the plants were tall. So this was actually capital T, and this was actually small t, small t. Now let us try to see what happens if capital T is cross-pollinated with small t, small t. Now you know the technique how to cross-pollinate it and how to find out the outcome. So let us try to do that. So if capital T cross-pollinated with small t, small t. So what are the alleles present here? Capital T, here small t and small t. So what are the various possibilities when they cross-pollinate? So these are the various combinations possible. So this will give you capital T, small t. This will also give you capital T, small t. This will also give you the same, and this will also give you the same. So if you see, all of them are heterozygous, but all of them have got capital T, which is the dominant trait, and that is why each one of these plant is tall. So now by looking at these alleles, what do you see? They inherited the tall trait from one parent and the dwarf trait from another parent. But since the tall trait is more dominant, so the F1 generation was all tall. So F1 generation was all tall. So now you understand the reason why F1 generation is all tall. But at the same time, you also see that each of these plants also have a dwarf trait which is hidden in them. The dwarf is not expressed because it is a recessive trait, but it is still present inside each of these plants. Now, what did Mendel do next? He self-pollinated the plants of F1 generation. So, which were the plants of F1 generation? They were these plants. So, when he self-pollinated them, so which are these plants? Capital T small t and capital T small t. When he self-pollinated them, he found that only 3 plants out of the 4 plants, I'm just talking about the ratio. So only 75% of the plants were tall and 25% of them were dwarf. So that time he was first surprised that how come the dwarf trait came back again. So now we know from where the dwarf trait came, because the dwarf trait was also present inside each of these plants of F1 generation. So now when these two are crossed, what happened? Now it has alleles capital T small t. This also has allele capital T small t. So these are the possible combinations, right? So this will give capital T small t, capital T capital T, capital T small t capital T, and small t, right? So this is a homozygous tall plant. So maybe this is capital T, capital T. There is another homozygous dwarf plant, which is this one. And the remaining two are heterozygous tall plant. So that is why these 3 were tall and this one was short. Right? So now you understand why Mendel was obtaining these kind of results. Now, after studying this, Mendel performed a second set of experiments. What did he do? Now he tried to check for more characters, because in the first case, what did he do? He selected, in the first set of experiment, he selected 2 plants with a contrasting trait, that is height. So now he thought that let me take 2 different contrasting characters. So instead of just height, he took 2 characters which were different in both the plants. So what were those 2 characters which he chose here? One was color of the seed and the other was shape of the seed. So every plant will give seed, right? At least pea plant will give seeds. So he chose one plant with a specific color of seed. He chose a specific color and a specific shape of the seed, while he chose another plant with a different color and different shape of the seed. So now there are 2 characters which were different in the 2 plants. And then he performed the same kind of experiment to see what was the result. So what he did, he cross-pollinated plants with homozygous round and yellow seeds and homozygous wrinkled and green seeds. So you see, this round and wrinkled basically describe the shape of the seed. Yellow and green describes the color of the seed. Right? So how was this denoted? How do you denote these? So let us have a look at that. So it is homozygous. So that means when I say round, that means it is going to be capital R, both the alleles are going to be identical. When I say yellow, both the alleles are going to be identical. Similarly, when I say wrinkled, so maybe round if it is capital R, wrinkled we can denote by small r. So wrinkled and green. So if yellow is capital Y, let us denote green as small y. So this is more convenient to denote it as in this way. So this is what he chose, right? So now we will see what was the output in this case. So what he did, he took round yellow seeds and wrinkled green seeds. So this was capital R, capital Y. This was small r, small y. Right? So if you see, these consists of these alleles. Right? So each of these is an allele. So now, so what is an allele here? RY. Because in this case, one allele should have one member of each character. Right? So what are the alleles present here? RY. Similarly, what is the allele present here? RY. You understand what is an allele, right? So in the previous case, each T was an allele because each T was denoting one member of a type of character. But here there are two types of character, color as well as shape. Right? So allele has to carry one member of each character. So these are the alleles. Now what are the possible ways in which they can combine with each other? Now the various ways by which they can combine with each other will give us some result like this: capital R, small r, capital Y, small y. So this was the F1 generation. So he found that in F1 generation all the seeds were round and yellow. This proved that the round and the yellow characters were the dominant traits. It means the round shape of the seed and the yellow color of the seed was the dominant trait. So this was the F1 generation. So in F1 generation he got heterozygous, round and yellow. So here also in a similar way, what did he do next? He self-pollinated the F1 generation, like how he did in his first experiment. So he self-pollinated this one. Now self-pollination of capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. Right? So he self-pollinated. So now if you look at this, what are the alleles that this can produce? What are the possible alleles that can be produced from here? So the various possible alleles that can be produced are capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the various combinations possible. These two, these two, these two and these two. Similarly in this case, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Right? Now, as we have done in the previous case, so what are the various options that are possible? Now, this first allele, so each of these. Right? Now, each of these can combine with any of these. Now, these can combine with this. This can also combine with this. Like this first one can combine with this. This can also combine with this. This can also combine with this. Similarly, this can also combine with this. Right? So, let me write down those here. So, what is the first option possible? RY, RY. So, what are the options possible? RY, RY. See, this RY combining with this RY. The next is RY, R, small y. Then the third option is small r, capital Y. The third option will be RY, small r, capital Y. RY, small r, capital Y. And the fourth option is RY, small r, small y. Right? The first one may combine with any of these. Similarly, the second one can combine with any of these. So that ways I'll write down all possible options. So this will be RY, R, capital Y. This can be R, small y, R, small y. This can also be R, small y, small r, capital Y. Or it can be R, small y, small r, small y. Similarly, the third one can combine with any of these, which can give rise to small r, capital Y, capital R, capital Y, small r, capital Y, capital R, small y, or small y, small r, capital Y, small r, capital Y, or small r, capital Y, small r, small y. Similarly, the last one can also combine with any of these, which can give rise to small r, y, capital R, capital Y, small r, y, capital R, small y, small r, y, small r, capital Y, small r, y, r, y. Right? So these are the various combinations that are possible in these cases. Right? So now let us see which one is going to be of what kind. The first one, if you see, so wherever you have the dominant trait present, so that trait will get expressed. So wherever you have a capital R, that seed is going to be round. So this has a capital R, this has a capital R, this has a capital R. So wherever you have a capital R, all those seeds are going to be round. right? So these are all going to be round seeds. Wherever you have a capital yellow, they are also going to be yellow. So they are going to be round and yellow. So here if you look at it, these are going to be the round and yellow seeds. So all four will be round and yellow. This is round and yellow. This is also round and yellow, right? This is round and yellow. This is also round and yellow. This is also round and yellow. Similarly, all those which have a capital R but a small y, they are going to be round and green. So these three are round and green. Again, those with small r are going to be wrinkled in shape, but they have a capital Y, so they are going to be yellow in color. And the one with everything in small letters is going to be wrinkled and green. So what was the result? So the final result was that there were, so the total outcomes were, if there are total 16 plants which were the result, then out of that there were 9 plants which were yellow and round. That means round and yellow. 9 plants, right? Then round and green. 3 plants, wrinkled and yellow. Again 3 plants, wrinkled and green. 1 plant. So this was the result that was obtained, right? So what was seen? It was seen that even though round and yellow, these two are the dominant traits, that doesn't mean that round and yellow will always go together. A plant with a round seed can also be green in color. Similarly, a plant with a yellow seed can also be wrinkled, right? So this was his second set of experiment. Now with the help of these two sets of experiments, Mendel arrived to the principles of inheritance. Now we will see how Mendel arrived to the principles of inheritance. So let us look at Mendel's observation. So what did he first observe from these two experiments? First was that the F1 generation displayed only one of the parental trait, and the trait which was displayed was the dominant trait, and the trait which remained hidden was known as the recessive trait. So the F1 generation expressed only one of the parental trait. For example, in case of the tall and dwarf plants, what was the F1 generation? It was all tall plants. So the trait which got expressed was the dominant trait, and the trait which remained hidden was the recessive trait. Now, based upon this observation, Mendel came out with the principle of dominance. So this was his one principle. What was his next observation? The hidden trait in F1 generation reappeared unchanged in F2 generation. So he saw that in F1 generation there was no dwarf plant at all, right? But again in the F2 generation, he saw the same dwarf plant. That means that hidden trait did not vanish. So hidden trait was also somewhere present in the plants of the F1 generation, right? So that hidden trait was also there. Also, he saw that the hidden trait did not got lost nor it got mixed up with something else. I mean, it didn't happen that that hidden trait got mixed up with the dominant trait, right? So the hidden trait was there and it was retaining its own identity. That is why in the F2 generation, the dwarf plant came up. It not only expressed itself, but it also expressed itself in its own original form. The same dwarf plant expressed itself. So he observed two things. First was the hidden trait was present in the plants of F1 generation, and the hidden trait was maintaining or retaining its identity. So on the basis of this, he gave the principle of segregation, wherein segregation means to separate out, wherein he focused on or wherein he emphasized on this fact that during the pollination, each of the gametes, they separate out from each other and they retain their identity as and when needed. So they do not get mixed up with each other. The third observation was that 4 types of plants were obtained in F2 generation in dihybrid cross. What is a dihybrid cross? Dihybrid, di means two, hybrid means mixture. So two contrasting characters. So in his second set of experiment, so Newton's first set of experiment was basically a monohybrid cross. That means there was only one contrasting character. Mono means one. And Newton's second set of experiments, the one with green and the one with round and yellow seeds, right? So that is a dihybrid cross. So in that cross, he saw that there were 4 types of plants which were obtained in the F2 generation. So he started with 2 types of plants, one with round and yellow seeds and the other one with wrinkled and green seeds. But when he saw the output of the F2 generation, he saw that he got plants with round and yellow seeds, he got plants with round and green seeds, he also got plants with wrinkled and yellow seeds and wrinkled and green seeds. So he got 4 types of plants, right? So what did he conclude from this? He concluded that he started with round and yellow, crossed them with green and wrinkled. So this is what he started with at the very beginning. So from there he got the F1 generation, which was round and yellow. When he self-pollinated the F1 generation, he got round yellow as well as round green as well as wrinkled yellow as well as wrinkled green. So this meant that each of these traits, that is round or yellow, they are independent of each other, right? It is not that wherever the seed shape would be round, it has to be yellow in color. It can be yellow or it can be green. So the shape of the seed and the color of the seed, they are independent of each other, right? So any of these combinations are possible. So with this observation, he came out with principle of independent assortment. So these were the 3 principles which were given by Mendel, and they together form the principles of inheritance based on which the entire genetics came up. Right? So let us now quickly look at each of these principles.
I hope this video was useful. If you have a feedback to share, do let us know in the comment section. And if you really enjoyed the video, do not forget to share it with your friends as well. I will meet you all very soon with a new video, with a new topic. Till then, take care. Bye-bye.